everyone. Welcome to today's PacBio webinar, Implementation of HiFi Sequencing Insights from Clinical Laboratories. We have an exciting lineup of guest panelists here today who will be sharing their experiences in validating and adopting PacBio HiFi long-read sequencing in their laboratories. My name is Nina Gonzaluto, and I'll be your host and moderator for today's panel. And before we hear from the speakers, we will have a pre-recorded introduction to PacBio technology by Sarah Kingen. Before we get to the speakers, I just wanted to review some housekeeping. So if you have any questions for any specific speakers or just for the panel in general, there is a questions module provided on your control panel. If time permits, we will try to address these questions during live Q&A. Otherwise, we will send the questions to specific speakers for follow-up. There is PacBio literature available in the handouts module as well. Please take a look at these. On-demand recordings for this webinar will be made available at a later date, and you'll get an email about that. And just a friendly reminder, please fill out the post-webinar survey. Really helps us put together this sort of programming for you. Our panelists for today's webinar are the following. We have Anna Lindstrand, who is the director of the Clinical Genetics Laboratory at Karolinska University Hospital in Sweden. We have Dale Muzzy, who is the Chief Scientific Officer at Myriad Genetics. We have Keith Nijkamp, who's the Vice President of Research and Innovation at GeneDx. And we have Lysenka Vissers, who is Professor of Translational Genomics from Radboudumc. We'll hear more from these panelists with an introduction after Sarah's intro. So with that, I'll pass it to Sarah to briefly talk about our technology.
Thanks, Nina. I'll spend about 15 minutes giving you an introduction to HiFi sequencing and how it's being used in clinical research. In case you're new to long reads, HiFi sequencing provides long and accurate reads of native DNA. Reads up to 25 kb are possible, with median read accuracy of 99.95% and the best coverage of the human genome of any technology. Long HiFi reads provide comprehensive variant calling and haplotype phasing without the need for trios. We sequence native DNA molecules, so you get methylation without additional library or sample prep. Compared to short-read NGS, HiFi reads are up to 100 times longer and just as accurate. PacBio workflows are familiar to any lab doing routine NGS, and we have products to support you from sample prep through analysis. Our automation-compatible nanobind kits for DNA extraction support high-quality DNA, perfect for long reads, from a variety of sample types, including blood and saliva. Our library prep kits support whole genome, targeted DNA, and full-length RNA preps, and can be run manually or with automation. We have multiple sequencers on the market, including the Vega Benchtop, which is easy to use and affordably priced, and our mid-throughput Revio system for labs running at higher scale. Lastly, our sequencers perform base calling on instrument using algorithms from Google Health run on NVIDIA GPU processors. This enables manageably sized files ready to run on variant calling pipelines deployed in the cloud or on-prem. For human genomics, HiFi sequencing enables you to access more of the human genome. In 2022, a team of researchers led by Sergey Nurk at the NHGRI finally published a telomere-to-telomere version of the human genome. In this paper, they assessed the ability of different sequencing technologies to accurately map to the new human T2T reference genome. They found that more than 99% of the human genome is accessible to HiFi reads. Illumina falls short, and their data is not able to map to more than 200 megabases of the genome, which is equivalent to the length of human chromosome 3. If you can't map to the reference, you can't call variants. Because of this superior mappability, HiFi reads enable a more comprehensive view of the genome compared to standard NGS. Short-read NGS is a great technology for detecting many SNPs, small indels, and CNVs. However, it can't do phasing, measure structural variants, or do methylation without special sample preps. As we saw on the last slide, NGS is blind to 200 megabases of sequence, which includes about 400 medically relevant genes in the so-called dark regions of the genome. With HiFi, you get SNPs, small indels, structural variants, and CNVs. You can detect challenging variants like tandem repeats and variants in segmental duplications. HiFi sequencing provides phasing and resolves haplotypes even without trio data. And lastly, methylation comes for free in all standard WGS and PureTarget libraries. So in summary, HiFi data delivers a new class of WGS, including all known variant classes, phasing without trios, and methylation without additional prep. Let's review some examples of recent high-impact studies that use HiFi sequencing. A recent preprint from the All of Us research program demonstrated the profound benefit of incorporating long-read sequencing into national biobank efforts and showed that long-read sequencing is a critical foundation for more equitable precision medicine. This was the first large-scale analysis of long-read sequencing from the All of Us program and provided a new framework for deriving genomic insights into complex structural variation, which has relevance to human health and disease. They analyzed over 1,000 individuals self-identifying as Black or African American. They identified nearly 300 SV disease associations across 226 conditions. And they found that more than 50% of these associations were absent from matched short-read WGS call sets. I'm including figure 4 from the manuscript, which highlights a notoriously challenging gene CYP2D6, where the authors were able to reconstruct haplotypes, including copy number variation and hybrid structures, and then call variants, which were classified with alpha missense by Google. A second paper I want to highlight was published in JAMA Pediatrics by clinical researchers at the Children's Mercy Hospital in Kansas City. And this study assessed the impact of clinical long-read sequencing as a frontline genomic assay for genetic disease diagnostics. The study included 235 participants aged 0 to 18 years. These individuals received HiFi WGS sequencing and were compared to an age and phenotyped matched control cohort of 513 individuals. Compared to the control group, HiFi WGS resulted in significantly more diagnostic findings in a significantly shorter time. The standard of care compared to long reads included inpatient genetic testing consisting of exome or genome sequencing, karyotype, FISH, chromosomal microarray, and targeted panels. Notably, there were fewer follow-up assays in the long-read cohort, meaning the HiFi genome resulted in a higher diagnostic rate with a more streamlined set of assays in a shorter time frame. And the figure on this slide shows that the diagnostic yield for long-read sequencing was higher, and the time to diagnosis was faster compared to other assays. So far, we've talked about HiFi WGS, but what if you need to sequence priority genes in a large number of samples at a low cost? PacBio also sells a targeted sequencing kit called PureTarget, which enables native HiFi sequencing at scale. This method is PCR-free and uses Cas9 to target genes of interest from as little as 1 microgram of DNA. We sell 2 panels for challenging targets, for carrier screening, and for repeat expansions. We have a manual and automated library prep workflows. On Vega, you can do flexible batching with PureTarget, processing as few as 8 samples and up to 48 samples per run. On Revio, with the 96-plex automated prep, you can process 100,000 samples a year on a single Revio. Compared to other targeted long-read methods, PureTarget has many advantages, which are shown on the right. You get epigenetic marks like methylation and no PCR bias or errors, which is a big benefit over PCR amplicons and hybrid capture workflows. Compared to targeted native long-read methods like adaptive sampling from Oxford Nanopore, the read depth with PureTarget can be hundreds or even thousands of reads on target. And as I said, the method is highly scalable with automation run on the Revio. We have 2 fixed panels available for PureTarget, including a 12-gene carrier screening panel and a 38-target repeat expansion panel. The carrier panel is designed to replace ancillary non-NGS assays used for tandem repeats like FMR1, genes with copy number variation like SMN, and genes with structural variants like F8. All of these genes can be run in a single long-read panel with a single prep. For the repeat expansion panel, we can sequence through long GC-rich repeats and get precise repeat length and motif sequences, methylation profiles, and deep coverage to characterize mosaicism. We have enabled custom panels so you can compile your own panel of genes from these existing designs or design your own targets. PureTarget can also be used to screen methylation targets like genes relevant with imprinting and to characterize engineered animal or cell models. Even chromatin accessibility screens are possible when combining PureTarget with FiberSeq. In July this year, Athena Diagnostics, a division of Quest Diagnostics and one of the largest testing labs in the US, announced the launch of an updated ataxia LDT that uses PureTarget and Revio in the workflow. And in our press release in September of this year, Dale Muzzy, the CSO at Myriad Genetics and one of the pioneers of the use of sequencing and carrier screening, and a panelist today, highlighted the streamlined workflow and the enhanced sensitivity and specificity of the data coming out of PureTarget. I'll conclude by sharing some of the updates on our two HiFi sequencers, the Revio and the Vega. As I mentioned, the Revio gives you HiFi at scale and is capable of processing 2,500 human genomes a year, which is great for PopGen projects, large clinical research, and as we said, carrier screening with PureTarget. We've already boosted the output of the system by 1.5x since last year. Vega is our most accessible system and is rapidly being adopted by new users, with 63% of the instruments being sold to new PacBio customers. It's a great system for doing targeted sequencing, RNA sequencing, and genomes. So what's coming in 2026? For Revio, we're adding 5-HMC to our methylation calling capability. We're introducing multi-use smart cells, which will dramatically reduce the cost per genome with a simplified workflow where the cell washing and reuse is done by the instrument. And lastly, we're bringing improvements to our Spark chemistry, which will enable cell multi-use, but also increase consistency. On Vega, we're adding the Spark chemistry so that you can go in with less DNA. Only 500 nanograms per HiFi genome will be required on Vega with Spark. And significantly, we'll be offering shorter movie times for short inserts like PCR amplicons, so you can complete your sequencing run in one shift. And lastly, we're adding 21 CFR 11 compliance to the Vega system. I'll conclude here with a little more detail on the updated Spark chemistry, which we're calling Spark Next. This will reduce the cost per genome down to $300 to $350, which means HiFi gives you not just the best, but also the most affordable HiFi long-read genome. We've also committed to supporting Revio and Vega through 2032. So as a clinical lab, you can be confident in investing in HiFi now. With that, I'll pass it back to Nina to introduce our panelists.
Thank you, Sarah. Okay, so now we're going to take a moment to introduce our panelists or have them introduce themselves. Please introduce yourself, tell us about your institution and what applications you're using HiFi sequencing for. I don't see Anna back on, but maybe Dale, do you want to start?
Sure, happy to. I'm Dale Muzzy. I'm the Chief Scientific Officer at Myriad Genetics. I oversee the R&D team that comprises molecular scientists, computational scientists, and the tech transfer teams that help transition the work into our operational lab. We serve multiple different parties in genomics, including oncology, reproductive health, and mental health, and primarily through genomics and DNA sequencing. So it's been a delight to work with PacBio.
Thanks, Dale. Keith, would you like to go next?
Sure, thank you. Hi, my name is Keith Nijkamp, and I lead the research and innovation at GeneDx. So at GeneDx, our mission is to be able to shorten the time to diagnosis for patients with a genetic disease. Currently, the average time to diagnosis for these patients can be 5 years, which can then often lead to 5 uninformative tests and cost up to $10,000 during this time. And so, I think as Sarah had highlighted, we're really excited about HiFi sequencing because it will increase our understanding of these challenging regions and can really reduce the time to diagnosis and will increase our diagnostic yield.
Fantastic, thanks. Lysenka?
Thanks, Nina. My name is Lysenka Visses and I work at the Radboudumc in Nijmegen, the Netherlands. We run for the Netherlands size, a relatively large clinical center with up to approximately 25,000 cases being tested for rare diseases for germline variants. We currently use approximately 10, 15 different workflows to detect all the relevant clinical variants. In addition to that, we have a huge benefit that we have an integrated division where we have research, clinic, and diagnostics all being present in the same place. So we can also very much rely on each other's expertise to make the most out of what we can get from long-read genomes if the genomes remain negative. So we also do a lot of follow-up to see how more we can benefit from all the variants that we can have identified.
Fantastic, thank you. Let's see, I think Anna was having technical issues. There she is. Hi, Anna.
Hi.
Briefly introduce yourself and what applications you're using HiFi for.
So I'm a clinical geneticist from Sweden who works now as the director for the clinical genetics and genomics lab here at the Karolinska in Stockholm, but I've also been heavily involved in genomic medicine, Sweden, rare diseases. And I think with long read and HiFi, we really had a national kind of project aligning very well with our local projects.
Great. Yeah. Thanks so much. Okay, so for the next portion of the webinar, we had some questions that we prepared for our panelists. Specifically, we are going to go through that. But again, if you have any questions, please feel free to drop in the Q&A and we'll try to get to them at the end. Okay, so we'll start off with our first question. This one is for Dale. Dale, Sarah mentioned in her talk that you guys use PureTarget for carrier screening. Can you briefly walk us through your legacy workflow? Just what are the limitations or challenges with other technologies? What motivated you to consider switching to long reads? And in general, what's your experience been like with PureTarget?
Yeah, sure. Happy to talk about that. Well, so I'd say our experience with carrier screening is kind of similar to what Lysenka said a minute ago, that like it is like one test we deliver to patients, but it has like multiple molecular workflows in the lab because we need to interrogate a large number of genes with short read sequencing, at least currently, for SNVs and indels, but then there are a lot of challenging genes that have very high carrier rates due to their molecular complexity, and that complexity that causes a lot of genome rearrangements and resulting pathogenic variants also makes short read sequencing very hard. So this includes repeat expansions, genes with pseudogenes, and so on. And so there's just a lot of operational burden associated with that: cost, time, retests, redraws. Like it's just most of our challenge with the assay is localized to a very small subset of genes that are hard. And then like PureTarget comes in and basically just makes those easy. So and you can do it in one integrated flow, which is awesome. So that's just, that's extremely exciting. So the, in particular, what motivated us shift to to long read sequencing was the, really just seeing that PureTarget could be fast enough, cheap enough, like multiplexed is huge to be able to look at multiple targets, but also a lot of samples at once. I think that's something that some of the technologies have missed. Like this isn't, like an academic type of project where we can, run fewer samples and kind of take a very long time. Like we have, thousands of samples coming in a day. And if like the platform can't withstand that, you just get steamrolled. So that's what motivated the switch. And the experience has been really, really good so far. I mean, honestly, it's kind of jaw-dropping, like just to see the data on these things that have been so hard for so long be just so clean. It's honestly a delight. So I'll stop there.
Great to hear. Thanks so much, Dale. So our next question is for Keith. Keith, your lab is also involved in really high volume genomic testing as you work at GeneDx. And you guys, I assume, use a number of different platforms. For those in the audience who are new to long reads, how do PacBio workflows compare in terms of automation, sample, library prep, bioinformatics, all of those when you're comparing short read sequencing and genotyping technologies? And how has the validation of HiFi assays been the same or different to what people are used to?
Yeah, so I'll start with the sample prep. To get the highest quality HiFi sequencing data possible, we would like to start with high molecular weight DNA. This is not something that most clinical labs who do a lot of short read sequencing have thought about. And so it does take additional work to develop and validate new DNA extraction protocols. One thing we've found so far is that the high molecular weight DNA extraction works really well with whole blood draws. While it's challenging with saliva, cheek swabs, and sort of dried blood spots, which are becoming more and more common and more popular sample types. And so as a high throughput clinical lab, we have to be able to handle all types of samples. And so, these are very important samples, especially for our really young patients. And so it's critical that we can get these samples validated. And we're working really hard on that, and I think the PacBio team's been really supportive of helping us try to solve this problem. But that's definitely something to think about with the HiFi sequencing. When it comes to bioinformatics and secondary analysis, this is an aspect that sort of overlaps pretty well with short read sequencing, with the exception that there are additional variant types that HiFi sequencing can detect with high confidence, but these have been either missed or just not as highly confidently called in the past with short read sequencing. Things like the large copy number variants, structural variants, paralogs, and repeat expansions, as Sarah highlighted. So these types of variants will require extra variant callers and additional annotations that we may not have at scale with the short read secondary pipelines. I can say that we've been very happy with the secondary analysis pipelines developed and shared by the PacBio teams, publicly. But the main thing that our teams need to do is port them into production pipelines and validate the callers for clinical testing purposes. And so that's the work that's ongoing now. And then finally, I can talk a little bit about the automation of sample extraction and library preps. And this is critical for a lab of our scale to be able to keep sequencing costs lower and scale throughput for many thousands of samples per year. And so this is something that will take extra effort, but fortunately we do have a lot of experience with existing automation and robotic systems, and we're working to adapt those to the HiFi protocols. And then maybe to go back to the high molecular weight DNA extraction, really our goal is to try to use one sample extraction protocol for all of our types of DNA sequencing. And one advantage of starting with high molecular weight is we can use it for all downstream processes. So this is something that we're really working hard on.
Great. Thanks, Keith. Another question for Dale. Besides the benefits of the PureTarget assay itself, how has moving towards a single HiFi sequencing workflow benefited operational aspects? I know you mentioned this before, but just more detail around validation burden, staff training, cost management, et cetera.
Yeah, sure. So I'll sort of piggyback on what Keith said, that just the ability to automate this, and we also have a very highly automated laboratory and have put the HiFi workflow basically onto kind of native automation. So it's great that PacBio sort of supports that either way. Either they'll sort of help get you started up, or if you have your own system, you can adapt their chemistry onto your system. So that has helped a lot. And we have kind of over time had development where we have multiple systems for different assays, and being able to consolidate that on one is definitely more efficient. In terms of like validation burden, like it is great as both a primary assay and as a confirmatory assay. It's simple to run. The staff have an easy time using it. And I would say honestly, like the like lab directors, for those who know them, or our lab like finding like giddy lab directors. Like, it's not usually a word I would use for it, but like seriously seeing like some of the data from PacBio, they're seriously like just so like happy, and that's nice to see. Last note I'll just make on validation is like, because we have used it as an orthogonal validation for other approaches, and like on each of them, PacBio is like the truth. Like, and you can anything you might see on some other assay, it's like PacBio actually like figures if there were a concordance, like the PacBio is right, and you can explain exactly what happened in the other assay that went wrong that PacBio got right. So it's just it has a very like distinguished role, I think, in terms of our validation. So yeah.
Great. Thank you. And then one last question for Keith here. As PacBio technology evolves, for example, Sarah mentioned costs are coming down, throughput's going up. When do you think the shift will happen from single gene or targeted long-read panels to whole genome sequencing? And what is it going to take for us to get there?
Yeah, I think the simple answer is, well, when sequencing cost per sample and throughput is comparable to the existing short read technology, being used in the clinical labs is when I think the shift will, completely happen. But, there is broad recognition that whole genome sequencing and HiFi technology can give us better answers, more answers, higher yield. However, at the current throughput and the capital costs and overhead, the costs are on the order of 10x that of short read technology at this point. But as the new technology comes out and we get lower costs, I think that will sort of precipitate the shift. I would also say that we're really excited about the PureTarget assay because of the ability to multiplex and really get the costs per sample down to what the comparable tests are for repeat expansions. And so, I see that as the bridge. That's sort of the opportunity to get sequencers validated in the commercial lab and really get our sort of lab set up and secondary pipelines, as I said, automation, DNA extraction protocols prepared so that when, the whole genome cost and scale approach the short read technology, we're ready to go.
Great. Good. So still a place for targeted and whole genome.
Yes.
Great. So we're going to shift a bit. So Lysenka, who's I believe very much a whole genome shop, but Lysenka, your lab has a long history with PacBio. You've become experts in using the technology in clinical research. Can you describe how your lab is going from HiFi for research towards more routine reflex or even eventually first-line testing for rare disease?
Thank you so much for this question. And yes, we are the very early adapters of using HiFi sequencing. But even before HiFi sequencing was HiFi sequencing, we already had Sequel 1 instruments and Sequel 2 instruments, which we at that time mostly used for the amplicon-based approaches for the difficult regions in the genome to test, where we would otherwise have multiple workflows up and running to find the relevant disease-associated variants. I think if we go back to approximately a year or 5-6 ago, we did our first genome-based, trio-based approaches for HiFi genome sequencing, where we, of course, had the index patient as well as parental samples to help us understand the variation and to interpret them, because at that time, larger scale data sets for the filtering to find those relevant variants was still lacking at large scale. And of course, we could then see how well the technology worked in a research setting. We could see if you would use these kind of cohorts that were negative after all of the routine tests, how they would then increase diagnostic yields. And we've also done this similarly in larger European scale projects like SolveID. And if we look at the whole total picture of that, I think we've sequenced over 500, 600, 700 samples using these HiFi genome sequencing approaches. For the last 2 years, I think we've shifted mostly to see how we can prove the utility of long reads for more routine-based testing. And we've used this again by similar kind of studies where we initially focus on the increase of diagnostic yield, but simultaneously at the strategy to increase efficiency in our lab infrastructure, making all of the other workflows redundant. Our first studies so far, again, show the same great output where we can increase diagnostic yield by approximately 10%. And even if we look at the most difficult regions in the genome, which we have used positive controls for in 100 cases, we can see that almost the most easy and most difficult variants in the genome almost become the most easy ones to identify using this very sensitive technology. More recently, we've looked at 1,000 cases where we have done HiFi sequencing in parallel to our standard of care to see how well they are able to replace current standards. And we can also there see that the results are overwhelming, and that we can even proudly say that to answer your question, when will we become a first-line test? We have actually started last week on October 20th to do this as a first-line test for a certain number of clinical disease entities. So we are doing it as a first-line test today. And of course, we are hoping with all that research and our experiences that we can gradually increase our throughput and move all of our germline-based testing to HiFi genomes shortly.
Great. Congrats on getting that started. It's fantastic to hear. Thank you for that. We have a question now for Anna. Anna, you've done some amazing work scaling HiFi sequencing at a national level with Genomic Medicine Sweden. Following up on Lysenka's questions, how far are we from HiFi whole genome as a first-line test? Are there certain diseases or phenotypes where this makes the most sense in your experience?
Well, thank you. Yeah, I mean, I don't think we're far at all. We went about this slightly different, I think, in Sweden from what Lysenka described from her center. But as I said briefly in the introduction, I think there was a sort of a joint interest in moving towards long read in all the clinical genetics centers in Sweden. And this aligned very nicely with our Genomic Medicine Sweden project that we started building in 2017, but where we now kind of have all the pieces of the puzzle in place. So we did a very small national study focused on chromosomal rearrangements when the first Revio was installed in Uppsala, and used that to also develop a national analysis pipeline and to train. So we got samples from all the different regions in Sweden, and also encouraged people locally to look at the data, to analyze the data, and that was published last year. But that was really just a very small startup to the big project we're doing now, where we're analyzing 1,000 individuals with neurological disorders. And we want these to be new samples that haven't been pre-screened with other technologies, because I think the key step now towards a long read first genome in the clinic is for us to be able to analyze that. And to do that, we need databases to filter out the common variants that we don't think are very important for rare germline disorders, but we also need to train the people who are doing the analysis. Now, if you're a smaller center and you're starting out, maybe your throughput will not be at the level that it is in my lab. But here, it's really, we're talking about, if we have hundreds of samples passing through being analyzed each week by super well-trained professionals, we need to see the different types of variants in the HiFi data, both to be able to trust the data, but also to be able to trust our colors and ranking models and interpretation possibilities. And we also need to know what is a bad run, like when can't we trust our data, when do we need to do a top-up, etc. So we decided in this project to do 1,000 in a year. We have 3 Revios in Uppsala, in Stockholm, and in Lund, so the sequencing is done there. We're using a national pipeline, and the data is interpreted by the local teams. And I think this is actually a really nice, it's like an implementation / training project, but it's also a research project, because we will obviously also be able to find eventually more things in this data. It's not really, I'm not saying that we're trying to kind of compare long read to short read. It's more about knowing what are the possibilities and what are the challenges in this type of data.
It's great. It's a big undertaking, but very exciting. I think that's a good segue to our next question. We're going to go back to Lysenka. Could you tell us more about HiFi Solves? I know you guys have been, one of the leading institutions for HiFi Solves. What have you learned from working with other labs who are also trying to adopt HiFi in their clinic research labs across Europe? And how has the experience been the same or different in different labs? I know, Anna was talking about within one country, but when we expand that a bit more geographically, what have been your experience there?
Well, thanks, and thank you, Anna, for already alluding on how well this works if you do this at national scale. And I think that that's also something that the Netherlands may perhaps also want to take a lesson on. And I think the lessons that you also described on sharing data and making it an effort in teaching and training, I think that that's also what HiFi Solves really does. So this global consortium is there to investigate best how to use HiFi genomes and technology in clinical human research and clinics, with over 20 participating institutions globally. We are there to share our best practices. We develop and share best common pipelines. We are shared the best ways to pipette manual DNAs, how to best practices and exchange what works across all of the labs, I would say. If we look at the output so far, I think if we compare the experiences of the different centers, I think I'm allowed to speak with one voice, that we all can say that it works very, very well. And it's not necessarily based on the location that you're at. It's a very robust and well-equipped infrastructure that if you know how to isolate your DNA properly, that everything else will almost automatically flow. The EMEA focus of HiFi Solves is mostly so far on the homologous regions of the genome, and also there we can already see that if we use those HiFi genomes, again, as said previously, all the data look great, and almost the most difficult variants to identify just readily pop up. You don't need an awful lot of experience to get that going, and I think that is what we can also see and share now on this. What additional very nice thing I think HiFi Solves at the moment is doing, and Anna already alluded on the absence of large-scale databases and finding the right repositories to have large-scale data sets available for reanalysis, and also there HiFi Solves offers a great help. It's already possible to search the databases and the engines for specific variants if you search just on the genomic position. But there's also a lot of data sets out there that you can request access to also learn from what HiFi Solves brings so far to the contributing institutions. So I think it's a global effort divided over different hubs to really learn and make not the same mistakes on the same places, but just to make sure that we get the best practices across as soon as possible to make HiFi genomes, or even in a targeted approach like the PureTarget ones, move forward as fast as possible for clinical implementation.
Great, thank you. I think the last question is for Anna, and it's, oops, not moving forward. Yeah, just more detail on what you mentioned, scaling HiFi at the national level. What are some of the biggest bottlenecks, and what are some of the accelerators in adopting HiFi across multiple labs within a national health system? Maybe you can help out the Netherlands as well with this question.
I do think, just sort of finishing up my database discussion, I think this is really a big reason that everyone at this point sees the need for collaboration. And we have all done our NGS tests in different ways. And I think Sweden's a small country. We're like 10 million total. We're regionalized. We have 7 healthcare regions with university hospitals. And it's just, if everyone was going to do their own, pipelines and then databases, that would just delay the whole introduction of the next kind of level of genomics into healthcare, which no one wants. What also happened was that we decided to use HiFi genomes for the genome of Sweden, which is part of the genome of Europe. And actually, we are working on the same pipeline. So we're using the same long read analysis pipeline for the population projects as we're doing for the clinical rare disease projects. And that means that these data can feed into each other, and especially that the population project can feed into the clinics and give us a healthy database as a backbone for our interpretation teams. So I think that was really key, and a reason that everyone was really on board with doing this, and it came quite timely. the bottleneck, I think, is kind of finding that balance between streamlining, like we're all working in this way. If we're developing a joint pipeline and we want this to be validated and also go into becoming IVDR certified, it is more work than if there's a new tool popping out from PacBio or another team that people want to test. You have to be allowed to do some of those tests, but maybe not too much, because when it comes to really putting people into like writing the code, it's better that we write it in the national pipeline, right? To kind of find that balance between trying things and adding the right tools to our joint pipeline. And so I don't think it's been a problem, but it does require us to meet and discuss and work on this. And some of the problems, I think, with the national project we solved already before the project started. So as I briefly mentioned, back in 2017, Genomic Medicine Sweden was launched as a sort of bottom-up project, the very opposite of Genomics England, where we were like, okay, we have these 7, university healthcare regions. Wouldn't it be cool if we could do genomics together and kind of help each other? And that has really developed over the years towards having a national informed consent for rare disease research, having an electronic web page where people can consent with their digital IDs. So having like that as a backbone has really enabled us to think about doing a national study at scale, I think. And then we, as I said before, we already started working on our national pipeline 2 years ago. And I think with the smaller project, enabling people to see the data, to realize how nice this looks and how great this really is functioning, has also helped. The other project that I didn't mention before was our undiagnosed project, the Undiagnosed Diseases Sweden project, where we also did a lot of different types of analysis, including long reads, and we had a national solvathon hackathon where we analyzed data together. And I think these sorts of events also bring people together and increase this kind of team spirit.
Great, thanks for that. Yeah, we big fan of hackathons ourselves. Try to get involved as much as we can. Great. So that wraps up the moderated portion. Before we move on to the audience Q&A, we're going to do a fun round robin. We have a little bit more time, so you can go over one sentence if you want, but maybe keep it quick. So for all of the panelists, just stepping back, what's the biggest impact that HiFi long-read sequencing has had on either you, your lab, or the people that order tests from you? Maybe we could just do it in this order. So Anna, Dale, Keith, and then Lysenka.
On the research side, I love structural variants. I really think that one of the biggest clear gains from this technology is being able to resolve structural variants and finding the breakpoints of the rearrangements. I did a study, and I know some of the PacBio teams knows that I did a short mini sabbatical, 5 weeks in Seattle last year, and I actually brought 6 HiFi genomes with me and analyzed them myself, and were able to identify the balanced inversions that we know from karyotyping were there. So, and as part of that study, we were actually able to find one breakpoint that disrupted the gene and that caused a monogenic disease in that unsolved case.
Fun sabbatical. Dale?
Yeah, I'd say for me, I'd probably focus my answer on the CYP21A2 gene. I mean, it's a very hard gene associated with congenital adrenal hyperplasia. It's on our carrier screening panel. I wrote a short read calling algorithm for that almost a decade ago that has served a lot of patients really well, at the same time been like just a large burden to the laboratory. And so seeing like how PureTarget and HiFi reads just resolve that and make it easy has just been, again, sort of shocking and wonderful to me, really good for the lab, and ultimately also better for patients in terms of getting them better and higher confidence results.
Awesome. Keith?
Yeah, so we've been using HiFi sequencing for research purposes for the past few years, and I think one of the biggest impacts we've seen is the ability to resolve indeterminate cases and uncertain cases that we find with short read sequencing, and then doing secondary confirmations, which are usually lab design tests, give us sort of either conflicting results or sort of uncertain results. And then when we're able to run those samples, in our research whole genome sequencing, we can often resolve them. These are, the paralogs, the challenging genes like Dale just described, but also repeat expansions. I think another exciting aspect of it is the methylation. It's really sort of a better understanding of how methylation can impact disease. And so, I think that's really sort of fueling research for us.
Fantastic. And Lysenka?
I can repeat what everyone else has said, but I'll try to capture it in that one sentence. So for me, it would be the one first line tier test, genomic base for germline testing has really become a reality.
Fantastic. Okay, one more round robin question to close this out. What's one piece of advice you'd give to a laboratory director who is considering validating and adopting HiFi for routine testing in their lab? And we will go in the same order. So Anna, kick us off, please.
Well, I think you need two instruments. You need to somehow, think about what database you will use for your analysis, for sure. And then, I think you can just get started for sure.
Straightforward. Dale?
Yeah, mine would be just that you have partners in PacBio. It's like the support from that team has been excellent, and it's hard to incorporate any new technology, and I think that the support provided has been wonderful. So view them as partners.
Thank you. Keith?
Yeah, I second what Anna and Dale said, but I would add, we need samples. We need samples with, these challenging variants to be able to validate in a clinical lab. It's a lot of these, like I said, they've either been missed or uncertain. And so to really clinically validate, especially if we think about new repeat expansion disorders, it's like we have to understand the pathogenic range. So having as many samples as we can find and share throughout the community, I think is going to be important.
Thanks. Lysenka?
I think my advice would be that the technology has now matured enough over the last years that it is a reliable and robust one, and that it has the sensitivity and specificity that at minimum is at par with the current standard of care for most of the variant types to be detected. So it is, yes, a struggle to move to a new technology, but it's no longer the technology that one should doubt.
Great. Okay, so we've got a number of questions that have come in through the chat, and Sarah is going to pose them to the panel.
Great. Thanks, Nina. Thanks, Nina. Okay, so the first question is, I'm going to start with Anna, but we can go around as well. So the question is, how do you think about coverage for HiFi WGS? For short reads, we usually think about 30x coverage, but is 30x what you are using? Does it matter the use case? If you're doing sort of a rapid genome and focus on diagnosing a case, is 20x sufficient? So how do you think about coverage?
So there are a couple of problems with this. I think we have to realize that we're still using a lot of repurposed callers from short read analysis, and therefore I am really advocating for 30x. I think at the moment our kind of clinical coverage is set at 70 gigabases of HiFi data, which will end up being around 23x median coverage. And with short read, we have a minimum of 26x, but aiming to go higher, obviously. So I think we are not quite sure where we'll end up. But if we, for instance, once we have callers that have really been developed for long read data and really, use all the signals we have in there, we may be able to go lower. But I think if you're an early adapter of clinical HiFi and you really want to make sure that you're not missing anything that we're confidently calling with short read today, I think that coverage is still important. I mean, for instance, small deletions. I mean, we are very, very used to finding them with short read, and we trust the signals there. And a lot of the labs have stopped running arrays. But I think that if you have small deletions with the breakpoints in repetitive regions, it could be, we don't want to run the risk of missing them.
Great, thanks.
Does anyone else have comments on?
Yeah, Keith. Yeah, Keith, do you want to maybe comment? And then if anybody else wants to chime in, feel free.
Yeah, I mean, I agree with Anna. I think we're running, primarily 30x. What I would say is that I just think we need a lot more experimentation on this, and especially with the regions we don't know that much about. I also think one thing that's important from our, high throughput clinical lab like ours is mosaicism. We do want to try to be able to detect that. And I think we don't know the read coverage that's really required for that yet.
Anybody else want to chime in from the panel?
I would like to add that it is perhaps slightly controversial just hearing Anna and Keith talking, that we perhaps should no longer start talking about coverage, but that we start talking about haplotypes. And I think that there is actually a huge benefit to go there, because sometimes even despite having only 6x coverage at a certain locus, we know that we have seen both alleles. And we also thus know that we have seen or not seen, if not present, the variant. So I think we need more insight in how well we want to detect haplotypes and the phased information, and how we can make better use of that information, and to talk about the raw coverage as a metric. We have done a first analysis on our 1,000 samples so far, and for all clinical relevant genes, we know that in 95% of the times we have seen 2 alleles. So whereas that does not exclude a very low mosaicism, I think it is fair to say that you can use that as a metric to say that you have actually sequenced the better part of the most functional, relevant clinical part of your genome for your patients. So 30x is what we currently use today, having kick-started last week. But that is simply because at the moment we cannot combine enough samples on a single flow cell. But we are, of course, hoping for Spark to come up soon so that we can do that as well. And I'm expecting that we will then actually go to a 20x coverage genome, if that is the metric to use, because we also much rely on seeing the haplotypes there rather than just the coverage.
Yeah, and I would say, even though at Myriad we're using it more in the PureTarget approach, like that, the depth does matter even in a targeted sequencing context, because it's very advantageous to be able to multiplex many samples together, and obviously the more you put in, your coverage on any one region will drop. So our minimum, I believe, would be, and this picks up on what Lysenka was saying, would be 10x coverage, but you have to see both haplotypes, and it has to be kind of clean and so on. I think 20x and above is kind of a much more comfortable zone, but yeah, again, the power of multiplexing is huge.
Nina, I think there's one more question if you want me to.
Yeah, let's do one more
question.
One.
Okay, so, and this sort of dovetails a little bit on the coverage conversation, which related to the tools that you use, and as those improve, maybe being able to tolerate slightly lower coverage. So again, I'll start with Keith on this one. Which analysis tools are you using, especially for STRs and paralogs, thinking about the variant calling and also the variant interpretation tools?
Yeah, with the variant calling, we're using the tools that have been developed by the PacBio teams, the bioinformatics teams, to be able to call both alleles and to determine methylation and mosaicism and length and sequence. So that's been really great. In terms of tertiary analysis at GeneDx, we've developed all of our own internal interpretation tools. And what's actually nice is once we get the calls and we're able to sort of confirm them, then I think the interpretation is the same as what we've been doing before, but just with new types of variants, we're going to have to, annotations. I think Anna had brought up annotations, and databases are critical. And so, that's what we're working on, is trying to build up our internal databases and use the external, HiFi Solves, DNA stack type of tools to be able to develop better annotations for that.
Does anybody else want to add?
I can add. I think we also leverage the expertise of PacBio for just the core identification of variants. But yeah, the core of it is the PacBio tools.
Anna, did you have anything to add?
No, I think we work.
Let's start with Anna.
Sorry, no, we work very similarly. We have been using the tools that are recommended, focusing on the variant types that we are interpreting today, but also now moving into kind of, okay, how do we want to use ParaPHIs, and how do we want to visualize these results in the best way, and how can we then also move on into kind of using the specific information from the long reads for our ranking model, and how we prioritize variants, etc. So there's a lot of development happening, and it's quite exciting.
I guess, Lysenka, any thoughts there on that topic?
No, same like the others. We use the standardized callers that PacBio supplied, and the only addition that we do with our in-house tooling is the annotation of our own local databases, etc., that help us prioritize the relevant variants. We have done analysis to see if we see more variants using HiFi genomes than using the current standard of care, comparing it to exomes and short-read genomes, to also understand how we are possibly going to overload our diagnostic lab with the interpretation of more variants that they would normally see, but that is not the same. That's not any different. It's exactly the same number of variants to analyze in a certain gene panel across the variant types. So that is very reassuring. The only other thing that I would like to mention is what we are currently doing in-house is developing the tools for the methylation-based signatures. Of course, many of the rare diseases are caused by a gene or a variant in a gene that leaves a methylation mark in the DNA, and that can be very instrumental to help understand certain variants of unknown significance. So this is what we're currently assessing. The current standard of care of that is mostly done by array-based epi-signatures.
Fantastic. Okay, very exciting work. So we are at time. We do have a lot of questions that we weren't able to get to in the Q&A. We will share these with our speakers as well as our appropriate PacBio team, and we'll try to get back to you all. But thank you all for attending. Thank you all to our panelists for sharing their experience and insights. It was very informative and interesting for us. Reminder, please fill out our post-webinar survey, and we will let you know when recording is available for this webinar. Thanks all. Everyone.